All righty, so today's lab is going to be on natural selection and how populations are going to evolve as a whole. Okay, so last week we looked a little bit at evolution. We looked at how evolution can be taking place, how we have different evidences of evolution in terms of convergent evolution, divergent evolution. We're looking at homologous structures and analogous structures and vestigial traits and fossils and things like that. So that's kind of the nitty gritty, but today we're going to be looking at different kind of mechanisms of how these populations are going to be evolving over periods of time. Okay. So this is going to be lab 12, our second to last lab for this week, or for our semester. Okay. So just as a couple recaps here. Is everyone able to see the PowerPoint, first of all? Let's start there. Let me make sure I'm not going too far ahead. Yeah, okay. Okay, so just as a quick refresher, our DNA is going to contain lots of genes. So genes, like I said, are going to be just regions of the DNA that are going to code for specific traits. Okay. In addition to DNA, we're also going to have messenger RNA. Okay. And inside that messenger RNA, we're going to have specific sequences of nucleotides. And then the last thing that we're going to have is going to be our proteins. Okay. And so the proteins are going to be made up of amino acids. Okay. So in order to kind of go from DNA all the way down to proteins, what we're first going to do is we're going to take our DNA, okay, our very, very large instruction manual, okay, we're going to transcribe it into a shorter, more concise mRNA molecule. Okay. Then once we have our mRNA is going to be translated to help us form the protein that we want to form. Okay, that protein can then either lead to producing different aspects of our phenotype, or the protein can be involved with different processes happening within the cell. It can be what determines different characteristics about ourselves. These proteins can be any number of different things. Okay? But all these proteins are going to come initially from genes. Those genes are going to get transcribed into mRNA, and then the mRNA is going to get translated into some proteins. Okay. Now, when we're looking at genes, we're going to have different alleles. So alleles are different forms of the same gene, or different variations of the same gene. And so, for example, we can have genes for eye color, like having brown eyes versus having blue eyes. So same gene would be the eye color, and then the alleles would be the actual color they end up being. Okay. Or we could have a gene for hair texture, and we can have a straight hair allele or a curly hair allele. Okay. And so on and so forth. Okay. So alleles just different forms of the same gene. Okay. Now, when we're looking at genotypes, okay, these genotypes are going to be classified as either homozygous or heterozygous. Okay. So homozygous is where we have two copies of the same allele. Okay, one allele inherited from one parent and the other inherited from the other parent. Okay, so for example, we can have two dominant A alleles that would give us homozygous dominant genotype, or two homozygous recessive allele, which would give us two recessive alleles here, homozygous recessive genotype. Okay. Or we can be considered heterozygous, in which case we have one copy of each allele. Okay, one copy of the dominant allele and one copy of the recessive allele. Okay. Now, as long as we have a copy of the dominant allele is always going to be the one that shows up. Okay. Now, in the case, and so the recessive one won't appear in the phenotype. In the case of simple dominance and simple recessiveness. So, for example, we have a condition called cystic fibrosis. Cystic fibrosis is a recessive condition, a homozygous recessive condition. So if someone is a heterozygote, they're going to be a carrier or heterozygous for it. Okay. And so even though they're heterozygous and they have the one copy of the recessive diseased allele, okay, they're not going to actually carry or have the cystic fibrosis phenotype, because they're going to have the healthy allele, okay, the dominant A allele in this case. Okay. So they're not going to show any sign of that mutation, okay, that leads to cystic fibrosis. Okay. So we can look at the phenotype, and so the phenotype is considered the outward physical appearance of that particular trait. In this case, purple flowers versus white flowers, or brown hair versus blonde hair, or straight hair versus curly hair, or whatever it may be. It does be the phenotype, whatever you're actually physically able to see on an organism. Okay. And then the genotype is the genetic makeup, okay, of that organism, okay, what's found in the DNA. And so if we remember, the DNA, the genotype will always determine the phenotype. Okay, so let's all review. Any questions before we go forward? This is all just kind of review from things we've talked about over the past couple of weeks. Any questions here before we go forward? Okay, so if there's no questions, we're good. So we kind of just reviewed everything what we talked about in the last few weeks. And so today we're going to be looking at how evolution can take place. Okay? And so typically, in order for us to see evolution, the easiest way for us to do it, or the most obvious way for us to observe evolution, is by looking at phenotypic changes. Okay? So actually, let's go ahead and backtrack. What does evolution mean from a biology sense? Someone tell me how we define evolution. Right. So Kimberly says a change in the population. Right. So a change in the population, I'm taking that just a tad bit further, a change in the population over time. Right. So the most obvious way for us to see a change in the population is to look at the changes in the phenotypes. Okay. So if we're able to observe a change in the phenotypes, let me put this in blue here. If we see changes in the phenotypes, that is usually going to lead us to be able to observe evolution. Okay. Now, in order for us to have a change in the phenotypes, what do we need to see a change in first? Someone tell me. The genotypes, right. So we need to see a change in the genotypes. Okay. Now, in order for us to see a change in the genotypes, what do we need? We need a change in the what? Alleles, right. So in order for us to see evolution, we're typically first going to observe a phenotypic change. That phenotypic change is going to be determined by a genotypic change, and that genotypic change is going to be determined by a change in the allele frequencies. Okay? So this is all going to start with the changes in the alleles in order for us to see evolution. Okay, so I'll just leave that there. Oops. Okay. So that's exactly what we see here. So for us to see an evolutionary change, that is ultimately going to be due to a change in the allele frequency. Okay, so when we talk about an allele frequency is just the proportion or just the kind of the ratio of that allele within that particular population. Okay? The proportion of that allele in a population. Okay, so how do we know if a population is going to evolve? Well, what we're going to do is we're going to be going based off of the Hardy-Weinberg principle, or the Hardy-Weinberg equilibrium. Okay? So Hardy-Weinberg equilibrium is a mathematical formula that helps us to be able to know if evolution is not occurring. Okay? This tells us if evolution is not occurring. And there's 5 criteria, okay, that are going to allow Hardy-Weinberg equilibrium to hold true. In order for Hardy-Weinberg equilibrium to occur, or to be true, for us to see that no evolution is taking place, we need 5 criteria. The first one is that there's no gene flow. Okay, gene flow is just another way of saying no migration. Okay, so there's no individuals migrating in or out of a population. Okay, that's number 1. Number 2 is that the population must be large. Okay, we cannot be dealing with small populations, because if we have a small population, then that population is going to be more susceptible to genetic drift and population bottlenecks and things like that, and like we saw earlier in class. Okay. The third criteria is that mating has to be random. Okay. So mating has to be random in order for Hardy-Weinberg equilibrium to hold true, for no evolution to be taking place. Number 4 is we cannot see any natural selection. Okay, so if there's no natural selection, that means that all of the genotypes have an equal ability to survive. They're going to have equal fitness across the board. Okay. And then the last one is that there cannot be any mutations. Okay. So all 5 of those criteria must hold true for Hardy-Weinberg equilibrium to be a thing, to be something that is accurate. Okay. For us to see no evolution occurring. Okay. Now, if even just one of these conditions is not met, then that tells us that the population is evolving because the allele frequencies are going to be changing. Okay? So for example, if we have a mutation, okay, If you have a mutation, we're going to be introducing a new allele into the population. If we introduce a new allele, all the other allele frequencies are also going to change. If allele frequencies are changing, that's going to lead us to having evolution. Okay. If we have natural selection occurring, then what that tells us is that certain individuals are not able to survive. So certain alleles are going to get removed from the population. Other ones are going to increase in frequency because they're going to be more advantageous. That's going to tell us allele frequencies are going to be changing. And ultimately, that's going to show us that evolution is occurring. Okay. If we think about migration, okay, if we have new alleles migrating into a population, okay, that again is going to change the allele frequencies. Okay. Allele frequencies changing, we have evolution. So if any one of those conditions is not met, that violates Hardy-Weinberg equilibrium, and that tells us that evolution is taking place in that population. Okay, is everyone kind of following me so far? Does this make sense? Any questions? Okay. All right, so looks like that makes sense. Okay, so when we're looking at Hardy-Weinberg equilibrium, there's two formulas we need to use. Okay, and I'll show you how to use these formulas in a second because this is pretty much what we're going to be doing for today's lab, okay, or this week's lab. The first thing we're going to do is we're going to be looking at the first equation, which is 1 equals P plus Q, or P plus Q equals 1. And the other one is 1 equals P squared plus 2PQ plus Q squared. Okay, so what these variables tell us is that P is going to give us the frequency of the dominant allele. Okay, Q is going to be the frequency of the recessive allele. Okay, so P is the dominant allele, Q is the frequency of the recessive allele. Okay, so if we have the dominant allele, the frequency of the dominant allele plus the frequency of the recessive allele, that should equal 1. Okay, that should equal 100%. Then the other ones, so P squared is like if we have two of the dominant alleles. Okay? If we have 2 dominant alleles, that is going to give us the homozygous dominant genotype. Okay? So P squared is the homozygous dominant genotype. If we have Q squared, that's like having 2 Qs or 2 recessive alleles. Okay? So that is going to be the frequency of the homozygous recessive genotype, okay, little a, for example. And then 2PQ is going to be the frequency of the heterozygous genotype. Okay? P being the dominant, Q being the recessive, that's going to be the heterozygous genotype. Okay? So one of these formulas, the P plus Q equals 1, is the allele frequency formula. Okay? P squared plus 2PQ plus Q squared equals 1 is our genotype frequency formula. Okay? So what I'm going to do here is I'm going to grab my handy dandy whiteboard. And we're going to kind of give a quick example of how to use this formula, because again, you will need to do this for today's lab and then for the assignment at the end of today's lab as well. So I'm going to stop this share here so you guys can see this in a bigger picture. Okay. So I'm going to go ahead and write the formulas again. Okay. So our allele frequency formula is going to be P plus Q equals 1. Okay. And then the genotype frequency formula is going to be P squared plus 2PQ plus Q squared equals 1. Okay. Is everyone able to see this okay? Yeah, okay, kind of blurry. Let's see. Let me see if I can do this a little bit closer. Okay, does that help? Yeah, okay. Let me do this a little bit further back here. Okay, so I'm gonna go ahead and just kind of write out what all these variables mean again. So again, P is the dominant allele. We have Q, which is the recessive allele. P squared is gonna be our homozygous dominant. Q squared is our homozygous recessive allele, and then 2PQ is the heterozygous genotype. Okay. Okay. So those are kind of what we're going to be looking for here. Okay. So let's say we want to, or someone give me an example of an animal. Give me one animal, for example. Someone come up with one animal. So a cat. Okay. So we're seeing this as a cat. Oh, and Brianna said, okay. So everyone's okay. We're going with cats. Okay. So let's say that we want to look to see how many cats have black fur and then how many cats have brown fur. Okay. Let's assume that brown fur is recessive. And let's just assume we're looking at wild cats out in the wild. Okay. Okay. So we go in the wild and we find that there's about 4% of brown cats that are, sorry, there's 4% of cats that are brown. Okay. So 4% of cats are brown. Okay. The other, so, and then we should be assuming that the other 96 should be about black. Okay. So we start off with our recessive trait. Okay, whatever we know is recessive. And so this is going to be how we're going to be looking at Hardy-Weinberg equilibrium. The first thing that we're going to do is we want to identify what our Q squared is. Okay. So I'll go ahead and just write that here. So identify Q squared. Okay. So if we know that we have 4% of cats that are brown, and we know brown is the recessive trait, what is our Q squared going to be? If Q squared accounts for the homozygous recessive individuals. Anyone have an idea? Try not to overthink it too. So repeat the question. So we want to identify what our Q squared is. That's the first thing we want to do. Our Q squared is our homozygous recessive individuals. Okay. So if we want to find what Q squared is, actually, let me say it this way. What is our Q squared in terms of a percentage? What is our Q squared? What percent of cats are homozygous recessive in our population? 4%, exactly. So our Q squared, I'll just kind of put this here. So Q squared is 4%, so I'll write that as a decimal, 0.04. Okay. That's our Q squared. That is step number one. Actually, let me do this differently. Okay. That is Q squared. Okay, now what we want to do is we want to try to identify the rest of these variables. Okay, we have our Q squared. We want to try to find our P, our Q, our P squared, and our 2PQ. Okay, so looking at our variables and looking at what we have, what is the next easiest variable to find? Should we find P, Q, P squared, or 2PQ based off of what we have already? What's the easiest one to find from here? Q, right. So we want to find Q. So how can we find Q based off of what we have already? Right, so Jared, we're going to take the square root, exactly right. So we're going to take the square root, square root of Q squared, take the square root of 0.04. If someone use your calculators on your phone or something, and let me know what you should get. What should Q equal? So it's not 0.02. 0.2. Right, good job, Hezzy. So we should get 0.2. Okay. So we've just found our Q. Okay. So the first step was to identify our Q squared based off of the information we have available to us. Okay. The second thing we're going to do is we're going to find our Q. Okay. And we're going to find our Q by taking the square root of Q squared. Okay. And then here we get 0.2 for our Q value. Okay. Now, what's the next variable that's going to be the easiest for us to find based off of what we have available to us? So Kimberly says P. Right. So P is going to be the next one. Okay. So the next thing we want to do is we want to find P. Okay. The way we're going to be finding P, actually, let me ask you guys, which equation are we going to be using to help us find P? The allele equation, right. The allele frequency equation, which is P plus Q equals 1. Okay. So I'll just kind of write this whole thing out here. So we'll have P plus Q equals 1. If we want to look for P, we want to get P by itself. We're going to subtract both sides by Q. We're going to ultimately end up with P equals 1 minus Q. And then, let's see, this is going to be too low. I'm going to push this over here. Okay. So we're going to end up with is P equals 1 minus 0.2, which was our Q, which we found over here. Okay. 1 minus 0.2 is what? .8. Exactly. Okay. So that's the third one that we were able to find. P equals 0.8. Okay. Now, based off our information, what's the next easiest one for us to find? What's the next easiest variable for us to identify here? P squared, right. So next thing we can do is we can find P squared. And so all you would do is you would just square your P. In this case, you would square 0.8. And what would we get? .64. Okay. So we would get 0.64 as our P squared. And then the last thing for us to do, just let me write this over here. Find P squared. Okay. The last step is to find our 2PQ. 2PQ. Okay. And so for the 2PQ, that one is relatively straightforward. I'm running out of space here, so I'll just kind of erase this top part. Yeah, I'm just going to erase this here for now. Okay. We're going to look for our 2PQ. Sorry, this is kind of all over the place. 2PQ, you would just take 2, multiply that by P, which we found was 0.8. Our Q, which we found was 0.2. And so what do we get here? Uh, 0.16 is close. So 0.16, you multiply the 0.8 and the 0.2. Right, we should get 0.32. Right. So 0.32. Okay, perfect. That's our 2PQ. Okay, so that is the order in which you need to do these problems, okay, to tell us or what we're looking for. And I'll show us how to interpret it in a second here. Okay. So first thing you want to do is you want to identify your Q squared, okay, which we saw over here. Okay, then we want to identify your Q based off of the Q squared. That's this step here. We want to find the P next. That would be this over here, which brings us over here. So our P was 0.8. We want to find our P squared. And the last thing we want to do is we want to find our 2PQ. Okay. Again, sorry, it's all over the place. I apologize for that. Okay, so that being said, this tells us what we should see if there is no evolution occurring. Okay, if Hardy-Weinberg equilibrium is holding true, these are the values we should see in nature. Okay. So based off of our calculations, we should see that 4% of the cat should be brown. Okay. We should see that the brown allele is present in 20% of the population. Okay. We should see that the black, the dominant allele, should be present in 80% of the population. Okay. We should see that 64% of our population should be homozygous dominant, and 32% of the population should be heterozygous. Okay. That's what we should be seeing based off of our calculations. Okay. Based off of Hardy-Weinberg, assuming there is no evolution occurring. Okay. So once we come up with all these numbers, then as biologists, we go out into nature and we actually look to see if we actually get those numbers. Okay. If the numbers that we actually see going out to nature, when we actually look at these cat populations, if those numbers differ in any way from the numbers we calculated, then we know evolution is occurring. Okay. If the numbers that we see in nature deviate from what we see here, okay, that tells us evolution is taking place. Then we just got to go out and then try to identify and figure out which evolutionary force is at play. Are we looking at natural selection? Are we looking at migration? Are we looking at genetic drift? Are we looking at non-random mating? Are we looking at mutations developing? Okay. If any one of those, or I guess if our numbers that we see in nature do not match up with what we calculated based off of the Hardy-Weinberg equations, that tells us that one of those evolutionary factors is at work. Okay. Does this make sense? Any questions here? Because this is a big one, and this is going to be the basis for our entire lab for today. Okay. What questions do we have? No questions. Okay. So again, the way you want to start this is you want to start with Q squared, go with Q, then P squared, and then 2PQ. What you cannot do is you cannot start with trying to identify P squared based off of what you have initially. Because P squared only tells you that you're dealing with the dominant alleles, and you have no way of knowing whether you're looking at, or whether a dominant expressing organism is homozygous dominant or homozygous or heterozygous. So you always have to start in this order. You go Q squared, Q, P squared, and then 2PQ, okay, in that order. If you go out of this order, you're gonna get numbers that are all crazy. The other thing that you can do that I forgot to mention previously is if you're not sure if you got your calculations done right, you can always check your work, okay, based off of the allele and genotype frequency equations. Okay? So if we want to check the allele frequency equation, we can take our P, which was 0.8, add it to our Q, which was 0.2, add them together, they should equal 1. Okay, 0.8 plus 0.2 equals 1. We know that one checks out. That's good. Okay. The other one, we're going to check that one as well. So P squared, which was 0.64, plus our 2PQ, which is 0.32, that's about 0.96. Okay, 0.96 plus our Q squared, which we had initially was 0.04. That would get us 1. Okay, so we know our numbers work out. Okay, so we know we did our calculations correctly. Okay, if they don't equal 1, then you know you made a mistake somewhere. Okay, you can go back and try to figure out where you made that mistake. Okay. Okay, any last minute questions on Hardy-Weinberg equilibrium? Any questions? Okay. All righty, so if there's no questions, we'll go ahead and go back to look at our PowerPoint. Okay. So is everyone able to see the PowerPoint again? Let me move this out the way. Yeah, okay. Okay, so that's how Hardy-Weinberg equilibrium works. Again, Hardy-Weinberg tells us what we should be seeing if there is no evolution occurring. Okay? If our numbers in nature do not match up with our theoretical numbers that we came up with through these two equations, then we know evolution is taking place. Okay, so that is the Hardy-Weinberg principle. Now, we're going to take a look at all the different causes of evolution. Okay, what things can actually lead to evolution? The first one is mutations. Okay, so mutations are pretty rare, but they can be really important because they're a source of new alleles into the population. And either they can be converting a dominant allele into a recessive one, or a recessive one into a dominant one, or creating a whole new allele out of something that wasn't there before. Okay? So they're going to be a source of new alleles by changing the DNA sequences. Okay? Now, the thing about mutations is that they are not goal-directed. Okay? Mutations do not happen in order to accomplish some goal. Okay? Mutations just happen, and they happen randomly. Okay? And so these mutations can either be helpful in terms of improving fitness, they can be harmful in terms of reducing fitness, or they could be neutral where they don't have any effect on fitness whatsoever. Okay, okay, those are mutations. The second process that we can have is natural selection. So natural selection can either act on existing alleles, and so that can either cause some alleles to become more prevalent from generation to generation, or cause some alleles to get removed from the gene pool depending on the environment and the habitat that's available and present, and depending on the resources and the limitations to those. Or they can actually act on the mutations as well. Okay? So if we have a mutation that develops, they can either act to spread the mutation if that mutation is beneficial for that particular environment, or it can eliminate the mutation if that mutation prevents fitness, prevents reproductive or survival success. Okay. The third thing that we can have is called genetic drift. Okay, so genetic drift is where we have changes in the population due to random chance. Okay? And so in genetic drift, we end up losing genes or losing alleles just randomly. Okay? And so this is going to be much more profound and much more easily seen in small populations, much more prevalent in small populations compared to larger populations. Okay? So in larger populations, because there's so many different genes and so many different alleles, genetic drift typically doesn't have that large of an effect in large populations. Okay? But it is more easily seen in small populations. Okay? So here's the same example we saw from class earlier today. If we have a population of 10 frogs, 3 of them are brown, the other 7 are green. Here we have 30% green frogs and 70% brown frogs. Okay, just in our 10 frog population here. If by random chance we end up losing 3 frogs and those 3 frogs happen to be brown, okay, then all of a sudden now we go from having 30% brown frogs to 0% brown frogs, and then 70% green frogs to now 100% green frogs. Okay? So we had some very dramatic changes in the allele frequencies. Okay? Okay? And that's because our population was so small. Okay? Now, if we had a population that had 100 frogs, and we lose 3 frogs die. Okay, so here we have 30 brown frogs compared to 70 green frogs, so 30%, 70%. Okay, let's say we lose 3 brown frogs, just like we did in the previous example. And then now we have 27 brown frogs out of a total of 97. And now our allele frequencies just move from 30 to 28%, and then the green frogs go from 70 to 72%. Okay, so the changes in the allele frequencies are not nearly as dramatic as they are in small populations. Okay. So typically what we see here is that when we're looking at a small population, one of the alleles ends up becoming fixed, meaning that all of the individuals in that particular population will end up having one of those alleles, and then all the other alleles are going to get eliminated. Okay. That typically happens in a small population, just like what we saw over here. Okay. Here we had a small population, we lost 3 brown frogs, and now we have all green frogs. So the green coloring became fixed. All the individuals in this population have it now. And all the other ones become eliminated. In large populations, that's not necessarily the case because there's so many individuals and so many alleles in the population, it's very, very difficult for alleles to become fixed or eliminated. So every allele pretty much will stay in the population. Okay. That's assuming the population size stays stable. Okay. Any questions on genetic drift so far? Okay. Okay, so there's a few different types of genetic drift. One of them is called a population bottleneck, and we'll talk about this on Thursday when we get to in lecture again. But population bottlenecks usually affect populations, typically large populations, sometimes smaller populations. But let's just say we're dealing with a large population. We're starting with a large population, and then we have some natural or man-made events that happens that significantly reduces the population size. So let's say, for example, there was a volcano eruption that wiped out a bunch of deer, or there was a flood that wiped out a bunch of trees, or a forest fire that wiped out a bunch of squirrels, okay? Or something like that. Or in this case, it can be hunting or overfishing. Those are some man-made kind of non-natural events. Okay? So these are very significant events that end up significantly reducing the overall population size. Okay? And so this becomes pretty significant because you initially start out with a large population that had a large number of alleles, a large variation in the alleles, and then at the end of this disaster or whatever the event was, okay, we end up losing a significant amount of the genetic diversity. Okay? And so we only end up with a small amount of genes in the resulting population, the surviving population. Okay? So here's a kind of an example here. So let's say we're starting with an original population here. We see all kinds of different alleles, and then we have some bottlenecking events, some major event that causes us to lose a large portion of the population. Okay? Well, the population that comes at the end of that, or that is able to survive that event, now becomes the actual population of the surviving population. Okay? So this now the survivors now become the entirety of your population. And so again, we end up losing a significant amount of our genetic diversity compared to what we initially had at the beginning. Okay? So our allele frequencies change dramatically, okay? And then our genetic diversity drops significantly. Okay. So this is pretty much what I just mentioned already. So these are natural events or human-caused events that kill a large portion of the population. And so the individuals that survive become the entire genetic structure of the resulting population. So that being said, all the genetic structure of the surviving population post-disaster is a lot different from the genetic structure of the pre-disaster population. Okay. Okay. And again, this usually will result in a loss of genetic diversity, a significant loss in genetic diversity. Okay. So here is a couple of examples. So cheetahs, and we'll talk about this on Thursday, cheetahs actually pass through a bottleneck event as well. So they're one of the major examples of animals that have survived a bottleneck. And so the first one that they experienced was about 10,000 years ago during an ice age. And so that ice age ended up killing a large portion of the existing cheetahs at that time. Okay. Now the cheetahs that were able to survive were just a few particular cheetahs. Okay. And the cheetahs that resulted there, that were able to survive the ice age, began to reproduce with each other, but the genetic diversity was so small that basically the cheetahs were all pretty much related to each other in some form or fashion. Okay. And again, that's going to be due to just a handful of parents in that surviving population. And again, we'll talk about that in more detail on Thursday, okay, in lecture. Okay. And then in addition to that, they also survived another bottleneck event more recently that was due to poaching. Okay. And so people were poaching cheetahs to kind of get their fur and their coats and things like that. That significantly caused the cheetahs' numbers to decline even further to the point where they almost went extinct. So cheetahs are one of the biggest examples of animals that have gone through a bottleneck event. Another one are the elephant seals. And so elephant seals were hunted to almost extinction, I think within the last century or so. And so they were hunted down to pretty much about 50 to 100 individuals, almost extinct before they started to kind of protect the elephant seals. And so the hunting acted as a bottleneck event for the elephant seals too. And where again, you had a man-made event that killed off a large portion of the existing elephant seal population, to the point where we only had a handful of elephant seals. Okay. Today, because of more protections, their numbers are kind of up to 130,000 or so. Okay. So they're kind of on the other side of that bottleneck event at this point. Okay. There's another type of genetic drift, and this is kind of a special case of bottlenecking, and this is called the founder effect. The founder effect is where you essentially have individuals of one population. You have a subset of those individuals leaving, okay, to now start a new population in another area. Okay. So they produce a new population. Okay. So those individuals who leave, yeah, Gilligan's Island, basically. Yeah, so those individuals that leave and basically colonize a new area are now considered the founders. Okay. Now with this, what ends up happening is the genetic makeup of the founders, okay, may be slightly diverse at that point, okay, but as they start to reproduce, okay, because there's only a few individual parents that are able to, make babies, okay, all the offspring end up being more related to each other than they otherwise would be in a larger population. Okay. So, for example, if you're dealing with these individuals here, and let's say these individuals are just kind of mating with one another kind of across the board, the individuals that the offspring that they're going to be producing are all going to be relatively more related to one another than individuals as like if you were to take Gilroy and Morgan Hill and San Jose as a whole. Okay. Okay. So that's considered the founder effect. And again, we'll take a look at that in a little more detail in lecture on Thursday. So a couple examples of kind of founders were the Baikal seals. And so these, I believe, I want to say are off the coast of Russia, but so off the coast of Russia and Siberia. And so what initially happened with the Baikal seals is that there were some seals that are these seals that were on Lake Baikal, which were mainly saltwater seals. Okay? They mainly existed in the saltwater area. Okay? However, the place where they were at, the particular piece of land that they were on, separated, okay, from the rest of the island, okay, and ultimately caused those individuals that were separated to found their own individual colonies, and now they became the freshwater kind of Baikal seals. Okay. So that's one example of a founder effect that happened with those. The other one are the Galapagos Island tortoises, the Galapagos tortoises, which are huge and massive. And so these ones also seem to have inhabited the Galapagos Islands as founders as well. So part of me wants to kind of ride one of those tortoises and hang out, but those are some massive guys there. Okay, so those are founders. So that's genetic drift. Okay. So genetic drift can happen in a number of different ways. Any questions on genetic drift before we go into gene flow and everything else from here? Okay. All right, so the next one is gene flow. And so gene flow is just another way of talking about migration. Okay. So in gene flow, what we have is alleles moving in and out of a population, okay, as a result of migration of those individuals. Okay. So for example, here we have population 1, here we have population 2, and we have individuals moving or alleles moving from population 1 into population 2. Okay. Once those alleles get there into that second population, that causes the allele frequencies to shift and to change, and now we end up having some evolution occur, or that can lead to evolution. Okay. So that's what we see here with the western deer population and the eastern deer population. Okay. So there's this passageway where the western deer can cross over into the eastern deer. As they do that, now the allele frequency in the eastern deer population is now going to change because you have new alleles entering the population. And so we end up with evolution resulting. Okay. Now, depending on the population, or depending on the species that you're looking at, gene flow is going to take place in slightly different ways. Okay. So in the case of animals, okay, animals can just move from point A to point B, on their own, okay, in order to produce a migration. Okay. In the case of plants, for example, plants, in order for gene flow to occur, their migration is going to be based off of their pollen, okay, the pollen that they're able to produce. Okay. So as they produce pollen, the pollinators like bees are going to come in, they're going to take that pollen and then transfer that pollen from that particular location, that particular region, into another region. Okay. And so that's going to be a way of delivering new alleles into a new area, okay? Allowing alleles to migrate or allowing gene flow to migrate among plants. Okay? So migrations and gene flow is going to happen a little bit differently depending on what kind of species or organism you're looking at. Okay. All right, that's gene flow. Okay, so those are all of the different mechanisms that can lead to evolution. Okay. Now that being said, now that we've looked at the different mechanisms, now we can kind of see like how is it that species are going to form? Okay, how are we going to produce new species? Okay. Well, the process of producing new species is called speciation. Okay. That's the process of forming new species. Okay. So speciation is going to be happening in a couple ways. The first way is by what's called isolation of the populations. Basically, you separate individual populations long enough that they eventually become their own individual species. Okay. So this can be done through geographical separation, or this can be due to adaptive radiation. We'll talk about those on Thursday as well. Okay. But basically, you just block the interbreeding process between two populations here. And you block them for a long enough period of time that they eventually diverge into become their own individual species. Okay. And then we end up with genetic divergence, where, like I said, after they've been isolated for so long, they eventually will become their own species, just due to a long period of isolation. Okay. And they develop different genetic traits. Okay. So we can have different types of species isolation. So we have pre-mating isolation here. And so pre-mating isolation types or versions of isolation are geographical isolation, where you have two populations being separated by some geographical barrier. You can have ecological isolation, where the species just occupy different habitats. You can have temporal isolation, which is just where we have individuals that don't mate at the same time. So the mating times differ, or they mate and reproduce at different times of the year or different times of the season. Or they can have behavioral isolation, which means that they're just kind of their behavioral patterns are so different that they don't recognize each other as similar species. Okay. So we'll go through a few of them. The first one is geographic isolation. So an example of this was the horned puffin in Alaska and in Iceland. Initially, these were one initial kind of overall species, but as some of the puffins moved, or kind of once some stayed in Alaska and other ones moved to Iceland, okay, they were exposed to different environments, okay? And each environment had its own selection pressures, its own selective factors. Okay? And so they needed to develop traits or evolve traits that allow them to be able to survive in that particular environment. Okay? So as a result, the horned puffin and the Atlantic puffin look slightly different from one another. Okay? They look still kind of similar, but they have different coloring patterns and different other kind of more structural patterns and structural traits, to the point where they do not even recognize each other as species that used to be the same. They're completely different species at this point, and so they're so genetically different that they just don't reproduce with one another. Okay. So that's called geographic isolation. Okay. The next one is ecological isolation, and so ecological isolation is where you have different organisms occupying different habitats. And so, for example, the fig wasps will occupy different figs. And so as a result, you end up with some fig wasps that evolve specific traits for specific figs that they're on, while other ones will develop other traits for the specific figs that they live on as well. Okay. And this ultimately led to having about 750 different species of fig wasps. So this is an example of ecological isolation. They just occupy different types of figs, and so they develop different traits that allow them to best live on that fig. The next one is temporal isolation. And so temporal isolation is where you have differences in the timing of reproduction. Okay, so for example, the American green frog and the bullfrog were once closely related. However, it got to a certain point where the American green frog only would mate and reproduce in the early summer, while the bullfrog would only mate and reproduce in the late winter or the late summer. Okay. So their differences in when they would go and mate was different, and so they never came into contact with one another, and eventually developed different traits and different adaptations, which ultimately led to their different characteristics here. And then the last one is behavioral isolation. And so behavioral isolation is where you have different behavioral patterns. Okay? So for example, the great horned owl and the barred owl, again, were once more closely related to one another. Okay? As they started to occupy different habitats, they started developing different mating calls. Okay? So the great horned owl's mating call sounded one way, the barred owl's mating call sounded another way. So because their mating calls sounded differently, they never recognized each other as things that they could, or as individuals that they could breed with. Okay? And so it eventually got to a point where they just started mating with their own, that they eventually became their own individual species. Okay? So these are what we call pre-mating isolation. Basically what this means is any different isolation patterns that happen before these two can come into contact and try to reproduce. Okay? So these ones are going to happen and prevent reproduction from even taking place at all. Okay. Okay, and so that is that. Any questions on what we've looked at so far in terms of how populations can evolve? Okay. So then what I'm gonna have us do then is we're gonna kind of work on lab 12. We're gonna work on the first part of it together. So we're not gonna really worry about writing this stuff down, but we are gonna kind of finish the video that we introduced last week. Okay, so here, where is it? Hold on, give me 1 second here. So what we're gonna do is we're going to pick up on the same video we watched last week in terms of what Peter and Rosemary Grant were doing. I'm going to include the video here. And so what you're going to do is you're going to watch the video from the 4:48 mark. Excuse me, hold on. Sorry, we're going to start from the 4:48 mark, and then we're going to watch until the 9-minute mark, and then we're going to discuss what we saw. Okay, so I'll go ahead and write that here. So 4:48 to the 9-minute mark. Once you get to the 9-minute mark, you're gonna come back and then either say done in the chat or back in the chat or whatever, so I know everyone's back and so we can kind of discuss what we saw in the video. Cool. All right, any questions on what we're doing so far? So again, we're just going to watch from 4:48 up to the 9-minute mark. We'll discuss what we saw, and then we'll kind of finish the video in pieces going forward today. Cool. All right. If there's no questions, go ahead and go for it. Go ahead and enter that chat, or you can mute yourselves if you're not already muted. I'll mute myself as well. And then at the 9-minute mark, we'll come back and then discuss what we talked about. Okay. So again, stop at the 9-minute mark and we'll come back. Okay, go ahead and go for it, and then we'll talk about it in about 4 minutes or so. All right, so let's wait about another minute, and then we'll get to discussing this. Okay. Okay. All right, so we'll go ahead and start discussing this. So I'm going to go ahead and pull up some of the questions from the lab manual. You don't need to write these ones down, but I just want to discuss this as a group. Starting with this first question. So based off of what we saw in the video, first question asks, describe the beak sizes of the medium ground finch population. Okay. So what kind of beak sizes did we have in the population here? We got some small ones, right? What else do we have? Right, we had small ones, we had large ones, we had medium ones, right? So they were all kind of in a range from small, medium, and large beaks. Now, what were those beaks useful for, okay, in this particular population? What do we think? Right, Kimberly, exactly right. So help them to open seeds, right? So if they had beaks that were smaller, the beaks that were smaller ended up being able to just break down or open up these smaller seeds, the smaller, softer seeds. Okay? Those that had larger beaks were able to break the larger seeds. Okay? So the size of the beaks essentially acted as a tool, like we talked about last week, acting as a tool to help them be able to open up the seeds and use the food or kind of be able to consume the food, which was their particular resource for survival. Okay, that's good. So B asks us, how did the population of the medium ground finches on Daphne Major change as a result of environmental changes? Okay. So I'll kind of like let that one kind of bleed into this next question here. So as the environmental changed, as we talked about earlier, there was eventually, yeah. So it has to mean you don't need to worry about writing these down. So we're just going to talk about them as a group. But in terms of these questions here, so let's say we have a drought. Okay. The drought that we talked about earlier in class today, where only the large seeds were available. I'll kind of draw a little graph here. Okay. So I'll just say, you know, beak depth, I'll just say 1, this will be 5, and then 10 will be over here, okay, as far as the beak size. Or let me change this a little bit. Let me make this 10, and then we'll say 20 millimeters on this side. That's a bit large, but let's just say for the sake of our example here. Let's say before the drought, do we think we had probably an equal distribution of the beak sizes, or did we think it was more of, you know, one side versus another before the drought? Yeah, before the drought, it was probably about equal, right? There was going to be small seeds, there was medium seeds, there was large seeds available. So there's going to be small beaks, medium beaks, and large beaks also available. So I'll put our before the drought in red here. So I'm just going to say, you know, there's about the same amounts for small beaks, medium beaks, and large beaks. Okay. Now, after this drought, where only large seeds were available, what do we think happened? Okay, do we think we had, or actually, let's just ask yes. What do we think happened after the drought where we had only large seeds available? What do we think happened to the beak sizes? Right, the birds with the small beaks would die off, right? So there's going to be fewer that have small beaks. So I'll go ahead and do these guys in blue. So after the drought. Maybe the medium ones might survive a little bit kind of here. And then the larger ones are going to increase dramatically, right? So they might be, way up here. So you might have a huge portion of beaks that have, or that are larger beaks, a huge portion of birds that have larger beaks than the smaller beaks, right? So that's our graph here. So let's say this is 1 millimeter, this is 10 millimeters beaks, and then 20 millimeter beaks. Okay. So after the drought, we expect to see a larger number of large beaked birds, okay, and then a smaller number of these small beaked birds, right? Okay. Cool. So that's our environmental changes there. Okay, so now what we're gonna do is we're gonna go from the 9-minute mark to the 11-12 mark. And again, we're going to continue that video. And then once you're done in the next 2 to 3 minutes, we'll kind of answer the next few questions here. Cool. All right, go ahead and go for it. And then in about 3 minutes or so, we'll come back and discuss this more. Okay. All right, so based off our video, what did we see? Okay, what did we find out happened as far as the drought? Let's start with the drought first. Was our data actually accurate as far as what we predicted with our graph? Do we predict an accurate change? Or did we see the same trend, I should say? Yeah. Yeah, so we saw the same trend, right? The large beak birds were the ones that were able to survive because they were able to eat the food that was available. Okay? The smaller beaked ones were not able to survive. Okay? Now, we also noticed that there was another event that happened, another environmental change. Okay, what environmental change took place in the, I guess in this part of the video? Right, so there was a flood. Okay? So there was a flood, an El Nino event that caused lots of flooding over the island. Okay? And so when that event happened, which seeds now became available? Small seeds or large seeds? Small seeds, right. So small seeds became available. And so again, we kind of saw natural selection occur again. Okay? So the ones that were, actually, let me ask you guys this. Which birds were now able to survive now that the small seeds were now abundant? The finches with small beaks, the finches with large beaks. When the when the small beaks were available, or sorry, when the small seeds were available, not the large beak birds, but which ones survived, or which ones were better able to survive this new environment? Right, the small beaked birds, right? The small beaked birds were now able to survive because now they now had an abundance of small seeds. Okay. So they had an abundance of small seeds, and then in the case of the large beaked birds, they were not able to survive as well because their beaks were too large to be able to use the small seeds. Okay. So the small beaked birds were able to survive, and so the pendulum swung in the opposite direction. Instead of having all kinds of large beaked birds now, okay, after the drought, then after the El Nino, okay, after the flooding, then now all of a sudden we had a bunch of small seeds, and so the small beaked birds now became a lot more prevalent. Okay. So now the small beaks became a favorable trait, okay. And so these were the ones that started to develop more often, okay. And these were the ones that were able to be seen more prevalently. Okay. Does this make sense? Cool. Okay. So the reason that Peter and Rosemary grants are so like dope is the fact that they were able to see natural selection not once, but they saw it twice in a very short period of time. Okay, normally in order to see one natural selection event and actually be able to record it, it takes years and years and years and years to be able to see it. At least 10 to 15, maybe 20 years, even long, sometimes even longer than that. Okay. They were able to see it twice in a span of like 3-4 years, which is incredible and something that doesn't happen. And so the fact that they were able to see it actually occur in front of them in real time, that was pretty incredible. And so, like I said, these guys are probably the heroes of evolutionary biology. Cool. Okay, any questions so far? Okay, so if there's no questions, we'll go ahead and finish off the rest of this video. So go ahead and watch from 11:12 all the way to the end of it, and then we'll answer this final question, and then we'll talk about what we're doing for the rest of the lab this week. Okay, go ahead and go for it. And then again, reply when you're done with the video. right, I think we'll wait about another minute or so. Okay. All right, so based off of our last video, let's answer this last question here. So last one says, how did one ancestral finch population give rise to 13 species, each with different characteristics? Okay. What do we think? Someone tell me how this happened. So Brianna says they went to different living environments. Right. So there was initially one ancestral finch population that lived in the mainland of the Galapagos Islands, right? Once there was one population that was established there, some of the descendants started to move to different islands. Okay? As they moved to different islands, they were exposed to different environments. Okay? And so when they're exposed to different environments, what happened? Natural selection, right. So they started undergoing natural selection and they started getting exposed to different environments, and those environments require them to have specific traits. Okay? So the organisms or the individuals that had those traits were the ones that were able to survive and reproduce. The other ones ended up getting removed from the population. Okay? And so we ended up seeing that those finch species ended up evolving in order to adapt to their particular environment. Okay, adapting. Let me say that differently. Adapting in response to the environment that they were in. Okay, so that's how we ended up with 13 different finch species. Each had its own individual environment. Okay, each one of the individual islands. Okay, each of those finch species that occupied those islands needed specific adaptations in order for survival. Okay, and so Kimberly, yes, this is exactly an example of adaptations. Okay? Traits that allow an individual to be able to survive best in that particular environment. Okay? And that's how we ended up with 13 different characteristics, or 13 different species, to the point where if you took those new species and you brought them back with the original mainland species, they no longer interbred with each other because they were just so different from that original mainland species. Okay? So that's typically how we classify different organisms as different species. Okay, their ability to interbreed with one another. Okay, if they're not going to interbreed with one another or produce viable offspring, and we'll look at this more so on Thursday as well, then we know they're completely different species. Okay. All right. Okay, so that's all we're going to talk about for this video here. Any questions on any other questions on this video before we go forward? Okay. All right. So the last thing I want you to guys do, and this is going to be the lab for this week, is we are going to do a simulation, okay? As natural selection simulation. Okay? So what you're going to do first, so is everyone able to see the lab 12 assignment here? Okay, cool. So the first thing you're going to do is you're going to go to this website. And so this website, virtualbiologylab.org/population genetics. You're going to go to that website. You're going to find model 1 that says PopGen Fish Pond, and then click Launch Model, and it'll open a new tab. So I'll kind of show you how that's going to work here. So let's, let me show you here. Oh, okay, hold on. Give me 1 second. Okay. Okay, so is everyone able to see the virtual lab page? Okay, cool. So you go to this website, virtualbiologylab.org/population genetics. You're going to go to model 1, okay, which says PopGen Fish Pond. You're going to click Launch Model, and it'll open up a new tab. And so when this new tab opens, the first thing that you guys are going to do, or this is actually, let me back here. You should see something starting to show up, a little simulation starting to show up here. It's taking a little while. Okay. So while this is loading, there's going to be a background section So it's going to be a background information session. I want you to read that. Then I want you to read the tutorial section. And then once you read the tutorial section, it'll lead you right into the experiment section. Okay. So you go to run experiments. Any second now. Okay. And then it'll show up with our fish population. Okay. So what you're going to do first is you're going to alter your initial size and the population demographics here based off of the instructions that are inside the lab assignment. Okay. So if we kind of go back to the lab assignment here, it'll tell you exactly what adjustments to make. Okay, so for part 1, you're going to alter the initial size and carrying capacity to 25, and so on and so forth. You're just going to follow this. It'll tell you exactly what to do step by step. Okay. And the description of what you're actually doing and what it means, okay, for your population. Okay. So we're first going to start with a small population of fish. Okay. And again, you just kind of read this over. You'll do what it says to do. At the end, or kind of right before you start your experimental design, this is what your experimental design should look like. Okay. Once you're done, once you have it set, you're gonna click the reset button up here. That'll kind of clear everything that's already happened in the meantime, and then you'll hit the play button, okay. And that will start your data collection. You're gonna click to data, and then that will take you directly into your data here. Okay. So I think for this one, what we're going to be doing, we're going to reduce the population size to 25, reduce the carrying capacity to 25, the mortality rate we're going to drop to 5, brood size we're going to drop to 5, and then migrant R allele proportions, we're going to drop that to 0. Little r, the homozygous recessive genotype, we're going to leave at 1. The other 2 we're going to put to 0.7, which means that they're going to have a lesser ability or lesser a reduced fitness than the homozygous recessive ones. Okay. I'm going to go ahead and hit the reset button up here. So I'll click that. It starts over. You click to data, and then it'll automatically start your data collection. Okay. And so it'll show you what's going to be happening in each generation with the individual alleles. Okay. Now, so this is where you're going to be using the Hardy-Weinberg equations, okay, which we saw previously. Okay. So you can kind of speed this up by clicking these little arrows here. So I'm going to kind of speed this up quite a bit. You're going to stop at every 20 generations. So you're going to stop at generation 20, okay, and then you're going to record the data in the tables in the lab. Okay. So I have an example that's also uploaded onto Canvas as well. Okay. So in activity 1, or sorry, in part 1, you're going to take your population size. You're going to write that in for generation 20. You're going to write the percent of the white koi fish, which is the homozygous recessive genotype. Okay. That's what we see here is 0.4. And add that in here. Okay. Then we have the proportion of the heterozygotes, the RR, big R and little r. That also comes out to 0.4 in this particular simulation. It may be different for you. And then big R, which we're going to put over here. Okay. So that's going to be the data that we see based off of what we're observing in our population. Okay. Then from there, table 2 is going to be this guy over here, where you actually use the Hardy-Weinberg equations. Okay. So you're going to take your percent of homozygous recessive, which was 0.4. Okay. That's going to become your Q squared. And then using your calculator, you're going to calculate Q, P, P squared, and 2PQ. Okay. So this is not in your lab manual. This is completely different. What was in our lab manual, we're not able to do. So this is completely different. So activity 2 in the lab manual, you're not doing. This is going to be our activity 2, okay, this simulation here. Okay. So you're going to kind of calculate all of these. You're going to make sure that your P squared plus 2PQ plus Q squared equals 1. In this case, it does. Okay, if it differentiates, if it kind of differs, like if it's 1.01 or 0.99, that's okay. If it's close to 1, that's fine. But if you're at like 0.8 or 0.7 or like 1.5 or 1.8, then you did something wrong. Okay. But it should be somewhere around 1. Okay. So that's going to be your P squared. And then you're going to determine if evolution took place in that generation. Okay. In this case, evolution did take place because our p-squared was 0.14 that we calculated using Hardy-Weinberg. Okay. But our p-squared for our homozygous dominant genotype frequency should be 0.2. Okay. We came up with 0.14, so we know that's significantly different. Okay. In addition, we also look at 0.47 for the heterozygotes. Here was 0.4. Again, that's significantly different. So because these two numbers are significantly different, that tells us that evolution took place. Okay. The numbers that we got using Hardy-Weinberg were different from what we observed in the population here. Okay. And so you're going to do that for every generation from generation 20. You do it for generation 40, 60, 80, and 100. Okay. So you're going to do that for the first one. Part one is going to be kind of organized that way. Part two, where did it go? Okay, part two, you're going to switch it up. And so in part two, what we're going to do is we're going to, again, be working with a small population, but instead the dominant phenotype is going to be better able to survive. And again, you're going to kind of write in what you find here and then list what you get for your Hardy-Weinberg numbers and then determine if evolution took place. And then you're going to do the same thing with a large population. So you're going to alter it again. Again, it tells you exactly what to do. And then do the same thing, generation 20, 40, 60, 80, 100, and do it from there. For the last one, okay, you're going to put in a screenshot of your final data collection page. Okay. You're going to answer this question here. In this final simulation, explain why the recessive allele does not disappear from the population. Answer that question. And that will be due, I'm going to say that it's going to be due on Friday, because this is a little bit more involved and a little bit more complicated. So this will be due on Friday. The last thing that's going to happen is there's going to be some hard. Actually, let me start with that. Does this make sense? Let's do that first. Are there any questions on what you're going to be doing for this lab? Again, everything is all laid out here, and it should be all pretty straightforward and self-explanatory. Okay. Are there any questions on what's going on? No. Okay. So if there's no questions, the last thing that is going to be due, this will be due next Tuesday in lab. Are you going to post this lab later? Yeah, so this lab is already posted up on Canvas. I just need to add a place for you to submit it. Yeah, so it is a little complicated. You kind of have to do kind of a little bit of interpreting the data. But all the instructions are pretty self-explanatory, so it should be okay. But if there's any problems, let me know. Feel free to email me and I'll try to get back to you as soon as I possibly can. So it is a little complicated, it is kind of involved, so that's why I'm having it due on Friday as opposed to being due today. Okay, so yeah, so I will put a place for you to submit your data, or kind of submit your data on Canvas. The last thing you're going to do is the Hardy-Weinberg practice problems. Okay, so these are in page 95 and 96 of the lab manual. Okay. So let me pull up the lab manual here again. Okay. So on page 95 and 96, you'll find these Hardy-Weinberg practice problems. And then what you'll do is you'll just kind of go through these, answer the questions as best you can, and then you'll be able to submit these on Canvas as well. So there's 4 questions. Like I said, these will be due next Tuesday. I will say next Tuesday at midnight. If these will be worth an additional 10 points. Okay, this is not extra credit. This is part of the actual lab, but this is going to be an additional 10 points in addition to the lab from today. Cool. All right. Sorry, that was a lot. Any questions on what's going on with this week's lab? Okay, so if there's no questions, you guys are free to go. And then I will see you guys on Thursday. Like I said, if you have any questions, feel free to email me and I'll try to do my best in terms of getting back to you as quickly as possible. Cool. Yeah, so Christina, this lab will be due on Friday. Yeah, and so activity 2 is on campus. So what we just looked at was activity 2. The lab manual is activity 2, we're not doing. Okay, we're gonna do what is on campus. All right.